Hello everybody. Let's get back into reviewing AP Bio. Last time we talked about mitosis and meiosis and the whole cell division thing. And this time we're going to be talking about genetics. Now, I'm going to give you guys another tip for studying and paying attention. When you are watching videos like mine, there is a specific little tip that some people give out, which is doodle with your right hand. You don't have to be drawing anything in particular, but the fact that you are doing something with your non-dominant hand, which for me is my right hand, helps you pay attention better than if you were just to be sitting down. With that being said, let's get right into it. Genetics. So, genetics follows this whole idea of Mendelian inheritance, which was this crazy guy from way back when who had a bunch of peas make babies, and he found out that traits are basically inherited 50% from mom and 50% from dad. And these traits are either put as a capital or a lowercase letter. And if you have a capital letter, that means you have a dominant trait, which is means that you only need one of these to be expressing a specific trait physically. Well, if you have a lowercase letter, that means that a recessive trait, that it is a recessive trait, and that two are required to express this phenotype. So what is a phenotype? Basically, you have this whole DNA thing. Whatever you have in your chromosomes, that is your genotype. That's your genes. Your phenotype is how you express that. So I might have a long sequence of genes or of DNA that codes me to have physically brown hair or longer fingernail growth than others. So that would be an example of a phenotypic trait. Now, another thing that you should know is homozygous is when you have the same alleles, which are these traits. So 2 capital A's or 2 lowercase a's or whatever letter it is. Or you are heterozygous, which is when you have different alleles. So you have a capital A and a lowercase a. Now, this isn't really talked about a lot in AP biogenetic questions. Whenever you see a dihybrid cross, you're not going to see this. But it is good to know just in general. Polygenic is when multiple genes code for one trait, while pleiotropic is when one gene codes for multiple traits. So that's your all for one for all, that kind of thing. So this right here is a dihybrid cross. We are trying to go for either a block or a bubble shape, right? A block is a dominant trait because it's capital letter, and a B or a lowercase b is a bubble shape. So the one parent has a capital B and a lowercase b, and the other one has 2 lowercase b's. So you take one B and you bring it down, so that's the B and the B, and you have your lowercase b and you bring it this way. So you have 1B, 1B. And through that, we can find out that, there is a 50% chance that their offspring will have a block shape. Now you take your lowercase b from this side, you bring it down right here, and you take your lowercase b right here, move it there, lowercase b right here, move it there. And that shows, there is also a 50% chance that it will be a bubble-shaped offspring. So you could say that the genotypic ratio from capital B to B, or capital B lowercase b to 2 lowercase b's is 1:1. And the phenotypic ratio of block to bubble is also 1:1. And this won't always be the case. There are some other ones, but this is basically how dihybrid crosses work. So you won't always have one gene that completely dominates the other. Sometimes there will be a bit of a blend, specifically with incomplete and codominance. So with incomplete dominance, if genotypes are heterozygous, the genotype is a blend of capital A and lowercase a traits. So if you had a red flower, which is dominant, and a white flower, which is recessive, if they blend together, they will make a pink flower. And one isn't actually really dominant or recessive in this case. It's just that it's almost as if both of them are dominant and they're both fighting for it. And when they do, they blend together. Codominance, on the other hand, is if a genotype is heterozygous, the phenotype is equally distributed, lowercase a, uppercase a. So if you had a red petaled flower and a white petaled flower, their offspring could be a flower or would be a flower with equal number of red and white petals, right? So it'd be red, white, red, white, red, white, red, white. That's basically that. Pedigrees are a way to trace inheritance of a trait over generations, and I'm going to show this through X-linked recessiveness. So we talked about just recessive and dominant traits. There's also sex-related traits, okay? So you have autosomal traits, which are the ones where it's not affected by sex, and sex-linked traits, which are the ones that are affected by sex. So whenever you have a square-shaped thing on a pedigree, that is a male, and whenever you have a circle-shaped animal or organism on a pedigree, it is a female. Which means that it will always be X and Y for males, X and Y, and for females it'll be X and X. If it is dark, that means that it has the trait that we're talking about. And another thing that you should know is that Y acts as a recessive allele. That's why X lowercase a and Y make this square act up, I guess, and gives it the trait. While this mom, which has a X capital A and an X lowercase a, does not act up because it has a capital A, which means it's dominant, which means the trait isn't being shown. Now, it is a 50% chance that the offspring will have a lowercase a or a Y from the dad, and a 50% chance of getting a capital A or a lowercase a from the mom. And from that, you have your offspring, right? For example, in the P generation, we had an affected dad and a not affected mom. And in the F1 generation, which is like the first offspring, you have affected offspring, not affected, affected. And this specific affected female, XA, both lowercase, commingle with an X uppercase Y, or an X uppercase A and a Y male, which means that it's not showing the trait. And if they were to blend together, no matter what, the female offspring will not show the trait because it is getting that X capital A from the dad. But if it has a Y, it is guaranteed to be a trait showed, it's guaranteed that the trait will be showed or expressed. As you can see, X lowercase a, Y, X lowercase a, Y. That's just how it goes. Sometimes you will have these like described as P, F1, and F2, depending on what like row it is in generation. And you might also have it expressed in Roman numerals and numbers. So the first row will be a Roman numeral 1, and the closest to the left will be a 1. So for example, this one right here is Roman 1, 1, and this one is Roman 1, 2. And row 2 would be a Roman 2, and row 3 would be a Roman 3. So this one is Roman 3, 1, this is Roman 3, 2, this is Roman 3, 3, right? That's just how it goes. Now you won't only have X-linked recessive traits, you might also have an X-linked dominant trait. So in this one, XA lowercase y, right? And this one is an X capital A and an X lowercase a. And it will spread out just like this. And something that you might have noticed is that X-linked dominant is the exact opposite of X-linked recessive, okay? See, as XA and X little a, or X big A and X little a, are not showing the trait. In X-linked dominant, it is showing the trait. So it just matters what you are shading in and not shading in, or what AP Classroom decides to shade in or not shade in. Now the next thing we're going to talk about is Hardy-Weinberg equilibrium, which is a way to quantify if allelic frequencies are shifting over a generation. And for a population to be in Hardy-Weinberg equilibrium, it has to have a large population, random mating instead of assorting mating, equal likelihood of reproduction for each genotype, so there's no natural selection, no immigration or emigration, which means nothing's entering or leaving the population, and there's no mutations. And if you want to be, the observant of you would realize, oh, well, isn't that like not possible for any animal at all? Yes, but you still have to learn it. So the Hardy-Weinberg equation comes in basically two separate equations, if anything. you have p plus q equals 1, and p squared plus 2pq plus q squared is also equal to 1. And p is equal to the frequency of a dominant allele, and q is equal to the percent chance of a recessive allele. And now p squared and p are slightly different in the sense that p squared is the percentage chance of having a homozygous dominant, so 2 capital letters, and q squared is the percent chance of a homozygous recessive, like percent frequency. And this is the same as the population showing the recessive phenotype. So if we go back to our block and bubble scenario from before, if you take your population of that animal and you just count how many bubbles there are and compare to that, the frequency of bubbles to the rest of the population is going to be equal to q squared, and you can square root that and you can find the frequency of the recessive allele. And that's kind of how you'd go about that question. It's actually relatively simple once you get the whole like algebra idea of it. It's actually not too bad. Let's talk about chi-squared. So chi-squared is a statistical test used to determine if data we collect, which is observed data, is significantly different from what we would have expected, which is our expected data. And this is done through sigma observed minus expected squared over expected. And now we would assume if everything is right in this card drawing scenario, which is what I have set up here, that the observed values would be the exact same as our expected values. That's what our null hypothesis is. This is like, oh, everything is going as it should be. Observed is equal to expected. Our alternative hypothesis is that the observed is not equal to the expected values. So that would be, what's it called? Yeah, just observed is not equal to expected. Now this will be given to you. This is called the alpha level, which is to like what extent you are confident that something will happen. So this is a 95% like confidence that, oh, so-and-so will happen. When you are doing these equations on a calculator, for example, it will ask you for degrees of freedom. Your degrees of freedom in this scenario would be 1, 2, 3, 4, right? That's the amount of rows, minus 1. So for this, our degrees of freedom would be 3 because it's 4 minus 1. And through that, we can calculate our critical value. You can look up more on just chi-squared if this is a bit confusing to you. But what you need to know is that we fail to reject our null hypothesis, or H of O, if the chi-squared is less than or equal to our critical value. But we reject H of O if chi-squared is larger than our critical value. Well, what does this mean? If chi-squared is really big, that means that something is happening where it's not equal, it's not what we expected. But if it's less than or equal to the critical value, then that means, oh, everything is going as it should be. I put in all these things right here. So 12 minus 13 is 1. Square that, divide by 13, gets you this number. Then I got this number. Da-da-da-da-da, it's just math. And I got a chi-squared of 0.77. If my critical value is 7.81, it is not large enough to cause chi-square, or it's not large enough to indicate that we can reject the null hypothesis. So that means that we fail to reject the null hypothesis, which means, oh, everything is going the way it should be if we're just drawing cards and putting them back in. That is everything that you need to know for genetics in AP Bio. Next video, we will be talking about DNA, and that will be what it's made out of, DNA replication, RNA transcription, RNA translation, translation factors, CRISPR for bacteria, all this kind of stuff that you may or may not see on your AP test. All I can say is I wish you the best of luck on your tests.